Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Resin, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for a speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Sierkstra from Thermo Fisher Scientific.
Good morning, afternoon or evening, and thanks for joining this webinar on our CaptureSelect Affinity Purification products. The presentation will focus on some of our new products just launched for different antibody formats. As well includes a case study together with Roche, but as well we'll start with a general overview on our technology. So the CaptureSelect Affinity products are part of the Thermo Fisher Scientific Bioproduction workflow. As you know, the Bioproduction workflow is comprised of upstream and downstream, and within Thermo Fisher Scientific and our Bioproduction division, we specialize in upstream cell line and media development, mixing cell culture and fermentation, as well as harvest and collection, bulk storage, and final fill. The topic of today will be the focus on purification, and mainly on the CaptureSelect affinity ligands and resins. As you know, as you're all industry experts, the global biologics pipeline is changing, right? So if you look to 2019 and the biologics which are in clinical development, less than 30% are MAbs. If you go back a couple of years ago, this portion of MAbs was a lot bigger actually. And of course for MAbs, the standard solution which people will be using is Protein A. If you look to all of the new biologics entering clinic and in clinical development, you'll notice that a lot of them are not standard monoclonals anymore, right? So they can be viruses, nucleic acids, complex therapeutic proteins with, for example, half-life extending protein modalities attached to it, fat fragments, T cells, and antibody drug conjugates. And actually these new molecule modalities all have purification challenges in the fact that they do not have actual platforms like MAbs have with Protein A, which as well then drives a need for additional tools to enable platform purification of these type of molecules. Because if you do not have a platform like in the case of monoclonals Protein A, then you end up in multi-step purification processes. And the limitation there is that if you have multiple steps in your process, and even if you would have, let's say, four steps in a process over here, which, let's say, 90% yield, right? Then still what you're doing, even although 90% yield per step doesn't sound too bad, you're throwing away almost half of the product which is made in cell culture in your purification process. So what affinity chromatography does, and has been demonstrated to do so, of course, by the extensive use of Protein A, is reduce the number of processing steps in your process, which increases the product yield, reduces your bioprocess development timeline, and thereby actually reduces the time to market and overall cost of goods for your biopharmaceutical. So with affinity purification products, we really are trying to simplify the biomolecule purification process by reducing the amount of steps in your process. Within Thermo Fisher Scientific, and again that is the topic of the focus of today, we've got two solutions to address that issue and actually to reduce the amount of steps in a process. One is our CaptureSelect affinity resin portfolio, which are unique tunable affinity resins for any biological, which results in a high purity in a single step, as well as our porous polished resins, where we've got a full portfolio of ion exchange and hydrophobic interaction resins. The uniqueness of the porous resins lie in the fact that the performance is actually independent of the flow rate. So unlike, let's say, typical agaroses, porous resins you can run at very high flow rates and still get very good resolution. As said, the majority of the talk will actually go and focus on the CaptureSelect resin portfolio which we've developed. So what are the CaptureSelect affinity ligands? Actually, they are based upon 15 kilodalton small antibody fragments, which contain 3 CDRs and 4 framework regions, which then have the ability to be tuned towards any target protein or virus particle of interest. Which means that with these 3 CDRs, we've got the ability to tune an affinity resin for any target. We can actually design the process based on the molecular target and process needs. So for example, if the molecule is pH labile and it needs mild dilution, we can actually engineer that into the affinity ligand. The production process is in Saccharomyces cerevisiae, so it's a fully animal origin-free manufacturing process. The technology is used in commercial purification processes, and as such, we currently have the largest affinity portfolio available on the market. The means by which we make these affinity products available for large scale is that we start off with actually generating pretty big libraries against any target molecule, which we then, through sophisticated ligand screening in high throughput mode, come up with ligands which have desired specificity, desired binding kinetics, mild elution built-in, and enhanced stability. After what we call work package 2, we would move on to work package 3, where we would do the prototype resin screening, finally ending up with a final leads in work package 4, and then ultimately we would turn this into a CGMP compliant resin by doing work package 5, which will mean that we will do process validation, leakage ELISA development, and finally compile the regulatory support file to be incorporated into GMP processes. This is our standard mode and operation for all of the work we do. So whether it's a general new product introduction we're launching, or whether it's a custom designed resin, we would go through the stage of work package 1 through work package 5, and then actually make the affinity resin available for general use. What we've done over the years is we've actually demonstrated this principle of affinity chromatography for recombinant proteins, antibody fragments in this case, and for example AAV. And this is really the power of affinity chromatography as depicted in this slide. So by means of the specificity built-in of the immobilized ligand, we can recognize a specific target compound and not the impurities, which you can actually see here loaded on the gel. We would bind the target to the affinity ligand and wash out the impurities, which is depicted on this gel over here, where you can see all of the impurities coming off. And then with a simple elution mechanism, which can be pH or alternatives like chaeotropes or other additives, we can actually then elute very nicely in one single step the compound of interest. So in this case, it's factor 8. In this case, it's an antibody fragment. And here you can see actually the 3 viral coat proteins of AAV coming off in your single step. Which means that by introducing an affinity capture for all biologics, you can reduce the complex steps of, let's say, a non-affinity capture mode into a very easy max 3-step process for all biologics. During the last, let's say, 15 years while we've been working on this technology and bringing products into the market, we've done so for antibody-derived therapeutics, the topic of the talk today. We have products on the market now for biosimilars, biobetters, and plasma proteins, viral vectors, and recombinant vaccines. As said, the products are currently used in commercial purification processes. So what we have is the largest CGMP compliant portfolio for virus particles and recombinant proteins. For example, this is an example with a customer where we've developed an affinity resin for TSA, so a hormone, which nicely in a single step gives a very high purity and a very high recovery for a pretty complex recombinant protein. Besides TSA, we as well developed products for FSA, HSA, so human albumin for fusion proteins, HCG, TPA, TSA, and human growth hormone. And that we actually launched AAV8-9X to purify adeno-associated virus, as said, cut see data from Genethon, where Genethon actually compared AVB cephorose from GE Healthcare, which actually has got a CaptureSelect ligand on it, together with AAV8, 9, and AAVX, where you can clearly see, which is the green bar, that AAVX outperforms all of the other products on a variety of AAV serotypes, which definitely demonstrates that AAVX displays an extremely broad selectivity to both natural and synthetic capsids. Another interesting product in the viral space is actually what we call the C-tag. The C-tag is a single domain antibody which binds to a C-terminal tag sequence, which is defined by the EPEA amino acid, which allows for one single step purification of C-tag proteins. In this white paper of Chemical and Engineering News, the Jenner Institute at Oxford actually describes the use of this C-tag in producing a recombinant vaccine, which was left on when going and entering in clinical trials. And they actually describe this as a breakthrough for recombinant viral vaccines, because again, it simplifies tremendously the production of these very complicated proteins. If you're interested, pick up this white paper on in Chemical and Engineering Use. Again, the advantage is you can really get high quality and pure protein in one single step, mild dilution, protecting the protein of interest, or in this case, the recombinant virus, which enables your high target purity and yield from these complex mixtures in the one-step process. Again, a really interesting recent article. So besides the fact that we've made products for biosimilars, biobetters, and viral vectors, the main topic, of course, is the talk that we have launched products as well supporting the development of new antibody therapeutics. And towards that end, these are more or less formats which are currently out there, right? With courtesy to Spies et al., which actually did this nice overview in molecular immunology back in 2015. These are the different species. And again, these are only the bispecifics formats, right, which people are actually working on, which actually as well demonstrates, right, that although Protein A is the gold standard for monoclonals, for all of these new type of antibody formats, they actually lack a platform to manufacture these type of antibody derived products. So what we've been trying to do with the CaptureSelect technology is focus on parts of the IgG where Protein A does not bind, right? So Protein A will bind to the CH2-CH3 interface. And what we've been trying to do is actually come up with resins and ligands which binds to different areas of the antibody or antibody fragments. So for example, one of the products we've launched is CaptureSelect Ch1 Excel. This product binds to the CH1 domain of an antibody, which means that you can, as a platform, now purify antibody fragments, because it will only purify the intact antibody fragment. So it will not bind free light chains in this case, which means that if you have overexpressed light chains in your antibody fragment, they will not be bound to the Ch1 Excel resin, which ensures that you can have a single-step purification for antibody fragments. Another product we developed is called Kappa XP, which is a recent launch beginning of January, which was specifically developed for antibody fragments if you do not have overexpressed light chains, but as well bispecifics when people are actually working with Kappa and lambda variants, and you can then use Kappa to, for example, pick out specific bispecific antibody formats. It will bind to the constant region of the Kappa domain, so it will pick up free light chains. However, it's got a pretty high dynamic binding capacity and has got built-in mild elution as well. The last product in this family is called CaptureSelect FC XL, which will bind to the CH3 domain of the antibody, which means it's been specifically developed when there are issues with protein A binding towards standard antibodies, but as well works perfectly well for FC fusion proteins, where you actually have got other modalities associated with the FC region. It's got a built-in mild elution as well. So the next topic is actually a case study which we did together with Roche. So this is courtesy to Jan Griesbach and Chris Teskey, who worked with us to implement the CaptureSelect FC Excel resin into a commercial next generation therapeutic manufacturing process. So the issue Roche had with one of their molecules is that one of their novel molecule formats didn't bind that well to protein A. They had no suitable alternative affinity resin available internally as a qualified GMP compliant raw material. And what they had was a process combining 4 different chromatography steps. And it was highly desirable for them to develop a process which was reduced to 3 steps, which could fit in their network facility. The disadvantage of the existing affinity resin they used, that it had a low binding capacity, it needed a low pH elution, which gave them some product stability issues. It had elevated fragment levels and an environmentally unfriendly regeneration solution. So what we actually worked together with Roche is we supplied them with the FC Excel when it was still, let's say, in early stages. And if you can remember still, FC Excel binds to the CH3 domain of the antibody. And what they did is they compared their existing affinity step together with an affinity B, affinity C, and a non-affinity step against CaptureSelect FC Excel, and compared it on low density, chop reduction, yield, the sum of the high molecular weight, the main peak, and the sum of the low molecular weight. You might notice still that the sum of the low molecular weight was a really big issue, which you can see over here, right, in the red with the high. So the only product actually which gave, let's say, a high low density, a good host cell reduction, a high yield, and a very low sum of the low molecular weight was FC Excel. So we worked with Roche and actually supplied them with a lot of different lots of the FC Excel which we manufactured, and they actually compared it on static as well as dynamic binding capacity to look to lot consistency, as well as yield, pool volume, impurities, and lead ligand. And what you can see over here in this slide, that the FC Excel and the different lots we've made had a very, very high consistency in terms of the different lots which were used, as well as a very actually good dynamic binding capacity for this specific monoclonal. It was higher than 35, between 35 and 40 in terms of capacity. Subsequently, what Roche did is they actually used the material to go from the pilot plant into their 3 GMP runs and compared it both in yield as well as the host cell proteins in the pool. And what you can see is that they had very good consistency going from pilot into GMP 1, 2, and 3. Similar and actually slightly better for the host cell proteins in the pool from pilot into GMP. The big advantage for Roche in this case was they could remove, because of the higher purity and the low molecular weight contaminants which were actually removed by introducing the FC Excel step, they could remove their process to 3 steps. Yeah, so they could move it from a 4-step to a 3-step process. Had a higher binding capacity using this resin, which improved their facility fit, and they had an improved impurity profile. I said no low molecular weight contaminants. A better pool stability because they could elute with the FC Excel at a higher elution pH, excellent scalability, and as well could return back to a more environmentally friendly regeneration solution. Besides, of course, the FC Excel, we as well do have Ch1 Excel and Kappa XP. I said Ch1 Excel was launched about 2 years ago and Kappa XP in January of this year. And over here on this slide, a summary of the Ch1 Excel data for platform, Antibody Fragment Purification. Again, this was published as well by Spooner et al., Amer Miyun, who is using this as a platform, a universal one-step process for correctly assembled fabs, published in Biotech and Bioengineering. And what you can see on this slide is a single-step fab fragment purification. So in this case, it's ranibizumab, so Lucentis, feeds from HEC 293 cells. Here you can see the load, the flow through, and the elution peak. Again, supported by analysis, where you can see that there's no light chain binding. This is the load with the light chain, this is the flow through, and this is actually the elution peak. And actually demonstrating that you do not co-elute the light chains. So Ch1 Excel is really a platform for antibody fragments, giving high dynamic binding capacity of your fabs, around 20 mg per mil, no co-purification of your free light chains, and efficient elution at mild pH. It will do every antibody fragment, so not only the Kappa family, but as well all lambdas. So it covers 100% of the Kappa and the lambdas. Next to that, we recently launched Kappa XP. Kappa XP is generic to 100% of all Kappa subtypes out there. So unlike protein L, it will do 100% of the Kappas. It's specific to human. It's got a high binding capacity for fabs, so for Kappa fabs, so actually higher than Ch1 Excel. And it's got a very high capacity as well for monoclonals. It's got built-in mild elution, good stability, excellent scalability, and as all of our products there, it's non-animal derived. So here you can see a competitive analysis of protein L, CaptureSelect, sold by GE, containing a CaptureSelect ligand, and the improved CaptureSelect Kappa XP. This is a 10% breakthrough analysis on polyclonal IgG. And what you can definitely see is that CaptureSelect Kappa XP has got the highest capacity for both fabs, in this case the blue bar, as well as the green bar, human monoclonals. If you look then on the right hand side to the elution efficiency, you can as well see that if you compare protein L to CaptureSelect to Kappa XP, you've got the highest elution efficiency at pH 3.5, right? Going from, let's say, 70% for IgGs with protein L and 36 for CaptureSelect. Kappa XP will give you almost full elution, which shows that Kappa XP is actually, whether you look to dynamic binding capacity or to elution efficiency, compared to protein L, CaptureSelect to Kappa XP, the superior product. Another example of Kappa XP data, but now for rituximab, you can see a nice purification for rituximab. It will pick up free light chains, Kappa XP, but it's got a very, very high capacity of around 50 grams per liter. Again, as well demonstrated here by another monoclonal biosimilar, where you can see the capacity against residence time. Of course, with any agarose, because this is on the agarose-based bead, you have a dependency of, let's say, residence time and capacity, but at 6-minute residence time, you're around higher than 50 grams per liter of capacity, which shows that if you are working with bispecifics and you actually are working with regular monoclonals, you can get very high capacities with Kappa XP. So the data which has just been shown are just in press. So the Ch1 Excel, the FC Excel, as well as the Kappa data are just actually being published by BioProcess International article in the October issue. So be sure, if you're interested, to pick up a copy of this. There's more data actually in the publication as in this webinar. So finally, we as well do have an active pipeline of new products we are developing. So this is actually what we call our pipeline slide, which depicts everything we have in development, from therapeutic proteins up to different antibody formats and types, up to viruses and vaccines. So for example, over here you can see products like GM-CSF, blood clotting factors, but as well host cell proteins we're working on. On the other antibody types, we are actually about to launch FC XP, which is the improved versions of FC Excel, which will have a higher capacity than FC XL, actually making it more, let's say, in line with current proteinase standards to be able to as well purify monoclonals. And on the viruses and vaccines, we are actively working on lentivirus and baculo and adenovirus to actually get those as a platform for GMP processes for these type of products. Again, as said, the lower we are on this slide, the less well advanced we are. But in any event, if there's interest in any of these products, we are always happy to either sample people earlier with products we have in development, or collaborate on any of the targets to get them from, let's say, stage 2 up to a bioprocess product. So this actually then reaches me to the final slide, which is that the CaptureSelect Affinity products have been demonstrated to give you a high purity in a single step, reduce processing steps, and actually seamless and gives you seamless upscaling for manufacturing, as depicted, for example, by the Roche case study. And it's I think fair to say that we are the only integrated supplier of affinity products with currently the largest product portfolio for affinity purification. And with that, I would like to thank you.
Great, thanks, Lawrence. So we do have some questions. As a reminder, just go ahead and type in those questions to the chat. So the first question is, what is the immobilization chemistry that is used?
So we can work with different immobilization chemistries, but currently the immobilization chemistry we use is epoxide. And we've got two different bead formats which we are commercially using. One is an agarose-based bead, and one is, of course, porous. And those are basically the two beads we use. Porous is extremely well suited for large molecules. So anything above 120 kilodaltons, we usually go for porous. Everything below that, we usually go for agarose. But again, it's a molecule-dependent approach.
And what are the measured DBCs of these resins?
So the dynamic binding capacities of any of the resins are usually translatable back to the molecular weight of the target molecule. Yeah. So for example, if you take FCXL, or FCXP, let's take FCXP, which is our product which we're going to launch in December. FCXP will have similar DBCs as, for example, the better proteinase have, right? If you then go for a antibody fragment, which is a third of the molecular weight of an IgG, you as well end up with approximately or slightly better than a third of the capacity of a protein A resin, because you basically are limited by then the molecular weight of the molecule.
Okay. And do you have data on ligand leakage?
Yes, absolutely. So all of our large-scale products, and of course, currently, as you've seen, we've got about 15 which are being commercially used. We, of course, as well do have, let's say, still 10 ligands which we supply through GE Healthcare. That all of the resins we use typically have leachate levels which are, let's say, approximately to what protein A is having. So it's usually below the limit of detection in all of our assays. So yeah, comparable leakage to what protein A would be.
And how are the CaptureSelect resins optimized for mild elution?
Yeah, so what we do have is, because we've been working with these products for about the last 15, 20 years, currently we do have a really good understanding of, let's say, the framework regions and what to modify if we actually want to, for example, further improve elution. However, a lot of the mild elution is actually coming from our screening procedures, right, where we actually, through our very extensive libraries, and our high throughput label-free detection and also screening systems, we will actually obtain those from the screening. Again, if the screening, let's say, needs improvement, then we have got a fairly good knowledge of what to change in amino acids in order to improve elution. The same is actually true for stability. So it's a combination of the screening methodology we use as well as site-directed.
Okay, and I think we have time for just one more question. If we didn't get to your question, we will pass this on directly to Lawrence and his team, and they will get back to you after this event. So the last question are these resins made in GMP conditions, and is there like regulatory paperwork?
Yeah, so actually these products are made to be used and suitable in GMP processes. So of course we cannot state we have got GMP because we're not making a pharmaceutical compound. But yes, they're up to the quality level of what is needed to be incorporated into GMP processes. Towards that end, for every resin we make, whether it is a off-the-shelf resin which people are buying, or whether it's a custom designed resin, we will make a regulatory support file which our customers are using to actually apply with the regulatory authorities. So that is all actually in our regulatory support files.
Okay, great. Thanks, Lawrence. And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.